Okay, so hi everyone. Thanks for joining us during the weekend. So welcome to our today's Immune Zoom seminar. So it's my great pleasure to welcome our today's speaker, Dr. Olivia Lu from UCSF. So so Olivia actually completed his undergraduate studied in Tongji University in Shanghai, China, and then pursue her PhD training in Beckman Research Institute in City of Hope. Following with the postdoc training in Genentech in San Francisco. So from 2019, she started her independent research program as assistant professor in Department of Neurosurgery in UT Austin, and then to 2022, I think. Yeah, she relocated her lab to Department of Neurological Surgery in UCSF until today. So currently, she's holding a position as associate professor in Department of Neurological Surgery in UCSF. So the main research interest in Olivia's lab is mainly focusing immunotherapy, tumor microenvironment, and crosstalk between the cancer cells and immune cells. Her research is focused on the investigating the molecular mechanism of immune suppression and invasion in brain tumor microenvironment with the goal to develop a novel immunotherapeutic for brain cancer. Um, Olivia's group actually has already identified the protein phosphatase 2A PP2A, which I think she's going to talk today, and then how it regulates immune suppression in T cell in multiple tumor models, including brain tumor. I think one of the highlights is based on these findings, phase 2 clinical trial of PP2A inhibitor is ongoing at the NCI for the treatment of glioblastoma. With her achievement, Olivia received numerous awards, which including Keystone Early Career Investigator Award and DoD, which is Department of Defense in U.S. Peer Review Cancer Research Program Career Development Award on 2020. So I think without further ado, thank you very much, Olivia, for joining us today. And then we are very much looking forward to your talk.
Thank you, Chen, for the introduction, and thank you, Xiaolei and Chen, to organize this amazing platform. And thank you for all the audience to participate today. And I know it's in China, it's Father's Day. And happy Father's Day to many audience here. So today I'm going to talk about like a protein phosphatase 2A, and I want to hopefully after my talk, I can convince you it's a really promising immunotherapy target. And really looking for some collaboration to develop a drug target this. So let me. So what is PP2A? It's a threonine and phosphatase, and so it has like three subunit. It's catalytic subunit C, and scaffolding subunit A, and regulatory subunit. Actually, when we talk about PP2A, because its B regulatory subunits is very diverse, has like 14 or 15 different B regulatory subunits. So, and it can cause like form different combinations. So actually, when we talk about PP2A, we meant like it's a whole enzyme combination, more than 60 different enzymes. Because of this, we face a lot of criticism, like the unspecificity when we target PP2A. So I'm going to talk about like, should we develop specific like PP2A based on the B or target A or C? I'll present some clinical data to we can discuss further. And also PP2A is widely expressed in all different kind of cells, like tumor cells or immune cells. And so actually when I joined, moving back to from industry to academia, actually I have untraditional path, spending 8 years in industry and before moving back to academia as tenure track faculty. So my goal was to develop a specific PP2A complex based on like the B regulatory signaling pathway or based on the cell types. In different cell types, they may play different roles, and then so that we can lead to more precision approaches to target PP2A. So currently, there is a PP2A small molecule inhibitor in clinical trials, and I have been working very closely with this company called Lixit. And there are several other like inhibitor targeting other phosphatase like SHAPE2 or SHAPE1 for immunotherapy or for cancer therapy. And so when I was still a scientist at AstraZeneca MedImmune, and actually through collaboration with a group at NIH, they already found that using this small molecule inhibitor targeting PP2A is enhance the tumor sensitivity to chemo or radiation. So the pathway is really nothing involved immune cells. It's more about DNA damage pathway. So when you inhibit PP2A in cancer cells and it upregulates P53, can induce apoptosis or cell cycle arrest, or it can induce like a phosphorylation of PLK1 and to induce the mitotic catastrophe. So and this is it suggests that PP2A is like very traditionally involved in DNA damage. So my background is cancer immunology. So then I was looking into whether PP2A play any role in immune components. Actually, back then, 2014, we started working on this project like more than 10 years ago. And then there's a recent paper, at that time there was a recent paper to do this unbiased screening, shRNA screening. At that time, there's not CRISPR screening yet. So they use the T cells and to use shRNA library to screen the T cells and then to use this melanoma sub-Q model, B16 model, and to see which target, if you knock it down, that can help the T cell enrichment to the tumor site compared to the control spleen. So they identify actually PP2A, one of the PP2A complex, so one of the top target. So that suggests actually PP2 in T cells may play some role to, if we inhibit PP2A in T cells, that can activate the anti-tumor immunity. That's why we tested this small molecule inhibitor and to combine with PD-1. And then I just show briefly the conclusion of the paper. So basically we found that it can synergize with PD-1 therapy in multiple type of cancers. More excitingly, it's in those cold tumors like brain tumor do not have too much infiltration of T cells. And then at that time, we focused on the mechanism of the T cells. If we isolate the T cells and differentiate them in vitro, and if you treat with PP2 inhibitor, it can promote them to produce more interferon gamma and then to promote them more Th1 differentiation. And then the downstream mechanism, so we found that actually PP2A modulate mTOR pathway. So if we inhibit PP2A, the mTOR pathway get activated. So and then it can promote the CD8 and CD4 T cell anti-tumor activity. But so because we use this the inhibitor to treat, so actually in vivo we target every type of cells, as I mentioned, tumor cells, all the myeloid cells. Actually in brain tumor, there's like predominantly myeloid cells in components. So we're trying to look into the more mechanistic study that I'm going to focus today for my talk. And based on those results, actually at NCI, that the clinical trials was initiated to use LB100 for the recurrent GBM treatment. It's definitely because it's glioblastoma. Just briefly some background. It's the most aggressive cancer and with overall survival of 15 to 18 months. Really, for decades, there's no therapy for GBM. And still the standard of care is radiation and chemo. So this really, those patients really need a novel therapy. And PD-1 clinical trials has failed recently for those recurrent GBM. So any novel therapy will be bring some hope for those patients. Um, and and then before I moved, this is actually this data is more recent. Like when we publish our paper, there are also some data from the just unbiasedly analysis of this more than 1,600 patients, and they were looking into which mutation this patient have can enhance the response to immunotherapy. And actually PP2A is one of the most dramatic one. And this is PP2A scaffolding subunit mutation, which is also like ubiquitously expressed in all PP2A hormone enzyme. As I mentioned before, the B is diverse, but A and C is the common one in all PP2A complex. So as long as you have this PP2A scaffolding mutation, you respond much better to immunotherapy, but not to other therapy. This is a very interesting data. And also in ovarian cancer patients, also very aggressive cancer, they do not respond to immunocheckpoint inhibitors as well. However, if you have this PP2A scaffolding mutation, it's dramatically improve the overall survival response to immune checkpoint blockades. So those are really like encouraging data to suggest that you target PP2A loss of this enzyme and then can sensitize to the immune checkpoint blockades. And those studies just recent data, and then meanwhile we're doing the mechanistic study, like how in different components in myeloid cells, if we genetic delete PP2A, what is the phenotype of myeloid cells? And today I'm also talk about if we genetic delete or PP2A in glioma cells, what is the phenotype? And actually, both of them are involved in STING and type I interferon signaling pathway. So the first part, we're going to talk about the role of PP2A in tumor-associated macrophages. So the reason to initiate this study, actually this is the first grant I wrote in my life when I'm moving back from industry to academia about the DoD grant. So I was very fortunate to get funded. That is very crucial for my career in academia. So the reason we started is because we saw the phenotype of the PP2 inhibitor, but the mechanism is not very clear because it target all type of cells. And then we want to look more closely what's the function of PP2 in each major components in the tumor microenvironment. And also there's a paper at that time showing in an infectious model that if loss of PP2 in macrophages, actually they produce more interferon in response to poly-IC or the SV virus infection. So I was wondering whether, because in tumor microenvironment, there's no virus infection, but those type 1 interferon has been like a very hot strategy to enhance the type interferon in cancer. I know there's some controversial, for example, in melanoma model, chronic type interferon may be not beneficial, but I'm talking about in those cold tumors, actually they have still there's a lot of strategy to enhance the type interferon in cancer, for example, tolerant receptor agonist or STING agonist or the oncogenic virus. Or even radiation, you can induce some double-strand DNA break and cytoplasmic double-strand DNA to induce the STING activation. For example, if you give glioma cells radiation or chemo, and then they can generate the DNA damage, and then you induce cytoplasmic double-strand DNA, and then to be a substrate of cGAS and to produce this cGAP and can be transported out to a myeloid cell to activate STING and to induce the type interferon production. But however, STING agonists failed very badly in clinical trials. So that suggests actually in the tumor microenvironment, there are some negative regulators of this pathway. So we're not that fully understand the negative regulation of STING in the tumor microenvironment. So then I was wondering whether hypothesize PB2A is a negative regulator of STING activation in myeloid cells in the brain tumor microenvironment. So the experiment we did, this first experiment, I'm very thankful to get this Lins M. Cree PP2A catalytic subunit knockout mice from Dr. Moore lab at University of Michigan. And we found that we get some bone marrow-derived macrophages, and we just did some RNA-seq, and we found that actually we treated with STING agonists, and we found that the knockout has much higher type interferon signature compared to the knockout. And we validate with those type interferon genes like interferon beta or CXL9 or 10 or SG15 is compared to the wild type, the knockout macrophages significantly increase those gene expression. And then we went to those mouse models. We tested different, not just the brain tumors, as melanoma or colon, and SB28 is very cold brain tumor model. And we also they all have reduced tumor growth in the PP2A knockout mice. And then they were wondering whether this depends on the TAPA interferon pathway. So we block the TAPA interferon using the interferon block the receptor use the antibody. And then after blocking the receptor of TAPA interferon, we do not see any difference between wild type and knockout mice. So suggests it is through the TAPA interferon. And then we were wondering whether it's through the cGAS STING. So we generate knockout, cGAS knockout as tumor cells and then implant to the host mice. There's no difference. So then this can help us to make the conclusion that the difference of tumor growth is dependent on the cGAS STING signaling pathway. And we also did the orthotopic model. Actually, the brain tumor microenvironment compared to the sub-Q, you can see in the sub-Q model, we just see the baseline that's different, but in the brain, it's way more immunosuppressive. There's no difference in the baseline. We have to give them radiation to elicit some double-strand DNA and elicit some baseline STING activation, and then in the knockout mice, we can see further improval of the overall survival. This is just some characterization of the tumor microenvironment that we found the more infiltration of CD4, CD8 T cells, and the macrophages become more MHC class 2 activation on macrophages. And also we did the single cell RNA-seq to look into more closely like different clusters. So the single cell RNA-seq data also suggests they have increase of the NK cells, CD8 and CD4, very consistent with our flow cytometry data. And then we're looking into the different clusters of TAMs, and we found that actually the most dramatic change is this is this interference cluster in a knockout, which is also consistent with our hypothesis that this is external modulation of the interferon type of interferon macrophages to initiate this difference of the tumor growth. And then so far we use is the PB2A catalytic knockout, and then we face okay, it's not specific. You're targeting every PB2A complex, so we're wondering whether we can screen like really identify a B regulatory subunit that regulate STING type interferon pathway. So we just did like tested all the PB3 regulatory subunit, and we found this one called stratum 4. If we knock down this stratum 4, it's a significant increase the STING activation. And what is this stratum 4? Actually, this stratum 4, do I have a picture? Oh, I think this stratum 4. So we confirm that if they knock out this stratum 4, the type of interferon STING activation is increased by qPCR and by Western blot, the phospho-STAT1 or phospho-IF3. And what is this stratum 4? Actually, this also like the first time I studied this stratum 4. So we did some research. Actually, there's some biochemistry studies about PB2A bind to stratums, including stratum 4, they inhibit this HIPO pathway. But this only all like those in 293 cells, the biochemical assays, and then they regulate the YAP test as a downstream. So as just HIPO and YAP test is like very important pathway involved in cancer or organ development, but not so much studies about them in immune cells. So now we first want to see whether this in macrophages, if we knock out stratum 4, they still regulate this HIPO, increase the HIPO activation. So, and then we found, yes, they indeed. And actually, we found actually this HIPO kinase is essential for STING activation. If we use this HIPO kinase inhibitor, MST1 or 2 inhibitor, we can totally block the STING activation. So this suggests actually HIPO activation is very important, critical for STING activation. And then what is the mechanism, how PB2A and stratum 4 act as a negative regulator of STING activation in the tumor microenvironment? So this is through a collaboration with actually my grad school friends, Zhipen, now a faculty at the University of Miami. His lab performed this co-immunoprecipitation assay. They found that in resting macrophages, actually PB2AC and stratum 4, they found very closely with this hippokinase MST1 and 2. And then if you give this STING agonist, they can induce this dissociation of this MST14 from this PB2A stratum 4 complex. But in the tumor condition medium, they always form this complex, inhibitory complex together, even you give STING agonist. And then we also, because HIPO's downstream is YAP test, and there are 2 papers actually from China showing that the cytoplasmic YAP can inhibit the non-STING, I think it's just type of interferon production in viral antiviral infection model by binding directly with TBK1 or binding directly with IF3. So we tested this, we overexpress this YAP mutant that do not have the TEAD transcriptional activity binding ability. And then we found that indeed this YAP overexpression of YAP can inhibit type interferon and STING activation. So and then we also found that actually in the tumor condition medium, or can trigger this YAP expression very significantly in the in macrophages. And also this is from the patient GBM microglia isolated from sorted microglia from GBM or sorted monocyte-derived macrophages from GBM. They all have very high enriched YAP score compared to their non-tumor microglia or blood or MDM from the blood. And we also show that the tumor in the tumor, the tumor conditioning medium can suppress the STING activation quite significantly, and then your knockout PP2A can reverse it, or your knockout stratum 4 can also reverse it. And then your knockout PP2A or stratum 4 actually can prevent the YAP expression in the tumor mediated by the tumor conditioning medium. So this is our paper published last year. So basically, we found that in the stratum 4 and PP2A form a complex that can inhibit HAPO activation, and then so further to stabilize the YAP test, and YAP test can inhibit the STING and the type 1 activation. So if we knock out this complex, this can be reversed and produce more type interferon to the tumor microenvironment. And then, so the next, I will talk about like the role of PP2 in glioma cells, because actually in GBM, like glioma cells and tumor-associated macrophage microglia almost are like more than 90% of the cells in the tumor microenvironment. So that's why we think, okay, we target PP2 in the tumor in the glioma associated macrophage microglia, we have like anti-tumor activity. How about in the tumor cells? Because historically there's some controversial about PP2A. And PP2 even was considered a tumor suppressor genes for many years because it can regulate AKT and CMYC. So we don't want to see, like, if we knock out PP2A in glioma cells and make them more aggressive, then suggest then we cannot use this as a therapy for patient. Or we need to think about more specific delivery of the drug if that's a case. So actually, when we CRISPR knockout PP2A catalytic subunit in this SP28 glioma cells, and then we see really dramatic improvement of the response to the immune checkpoint blockades. You can see this SP28 model is very cold tumor model. They don't have too many T cell infiltration, and they do not respond to immune checkpoint blockades. Like PD-1 alone do not do anything to this model. And this is very similar to the ovarian cancer patient data I just showed at the beginning. Like if they have this PP2A scaffolding mutation, also expressing every PP2A complex, they respond to the immune checkpoint blockades significantly for those very aggressive ovarian cancer patients. So this is very encouraging data, and then we're looking into more mechanism, and then we found that actually if you knock out PP2A in the SP28 cells, but we did multiple on GBM cell line, like including human, and then they dramatically like, increase the MSC class 1 expression on those cells. And as you can see, without the wild type GBM cell line, they do not express, they're very cold, they do not express MSC class 1. Almost baseline is like 0. And then this can increase to like quite dramatically. And then we're wondering whether this is dependent on type interference. So we block the type interference signaling, and then we can like reduce the MSC class 1 expression in the knockout. So this suggests that indeed the increase of MSC class 1 expression of in the PP2 knockout is dependent on the type and the furin. And then so we also wondering, okay, if they express so much more MSC class 1, whether they help them to be like enhance the killing mediated by the antigen specific CD8 T cells. So we overexpress over in those tumor cells and in wild type and PP2 knockout, and then we co-culture with dendritic cells, and then we put the so we first test whether they can activate dendritic cell more, and indeed and then we put the CD8 T cell in OT1 T cell and to measure the OT1 T cell proliferation. And we indeed find that the knockout glioma cells has increased ability to increase the proliferation of the antigen specific CD8 T cells. And then we're also looking into some mechanisms like how the MSC class 1 or type interferon was increased in those GBM cell lines. So because in cancer cells, they are involved in DNA damage pathway, then we was looking into the DNA damage pathway. They did have increased the gamma H2X and suggest that they have increased DNA damage unrepaired. And then also because we see more type interferon like signature, and then we see more MSC class 1, and we were wondering whether this depend on the CGAS STING pathway. So we did the cytoplasmic double-strand DNA staining, and indeed we found that in the knockout mice, there's significantly increased accumulation of those cytoplasmic double-strand DNA. So that can be recognized by the CGAS and then to activate STING to induce this downstream type interferon signature. And then we just also seen some immunogenic cell death markers and to show that those PP2A knockout GBM cells, they have increased immunogenic cell death. And that also has been shown to activate the CGAS STING type interferon pathway. So I think I'll stop here and to answer any questions. So I want to thank my team and particularly Isha, who just is going to join medical school in Duke, and Oishka is a medical student at Hopkins. And also thank my collaborator Vincent Ho, and also my other collaborators in US or UCSF.